Hi everyone, thank you for your interest in the poster titled A Validated Hybridization LC-MS Method for Quantitative Bioanalysis of HTKIT, vivo morpholino antisense nucleotide. Presented at the 2025 AAPS PharmSCI 360 conference in San Antonio, Texas. My name is Mingluan Chen. I am the principal research scientist at the Altasciences, and it will be my pleasure to discuss our research with you today. Vivo morpholino are a type of conjugated PMO. The structure is highlighted in Figure 1. These molecules can enhance cellular uptake and stability, making them essential for gene function studies, disease monitoring, and the treatment development. To support PK evaluation, sensitive and selective bioanalytical methods are essential. However, conventional LC-MS approaches face limitations due to the inefficient extraction and the poor chromatographic resolution, largely driven by strong electrostatic interaction originating from cationic vivo morpholinos. Our goal is to develop and validate a quantitative hybridization LC-MS assay for HTKIT, octaguanidine conjugated vivo morpholino in mouse serum over an analytical range of 1 to 250 nanogram per ml. As shown in Figure 2 and the methods section, the workflow involved digesting mouse serum samples and hybridizing them with the biotinylated capsule probe. The analyzed probe complex was formed, washed, and thermally denatured to release HTKIT. LC-MS analysis was performed using a SciX 6500 plus triple quadruple MS in positive ESI MRM mode. Detailed LC-MS parameters were provided in Table 1 and 2. As shown in Figure 3, we achieve clear baseline separation between HTKIT and the internal standard using 25 microliters of mouse serum. The chromatograms for blanks, the LLOQ at 1 nanogram per ml and the ULOQ at 500 nanogram per ml shows that the method is highly specific and provides a linear response across the range. Both HTKIT and the internal standard have sharp, well-defined peaks with excellent single-to-noise ratio and no interference observed in the blank samples. The calibration curve ranging from 1 to 250 nanogram per ml showed excellent linearity based on the peak area ratio of HTKIT to the linear PMO internal standard. Precision and accuracy were confirmed across 4 QC levels in 3 batches with 6 replicates per concentration per batch, all meeting the acceptance criteria summarized in Table 3. Matrix effect was evaluated using 6 independent lots of mouse serum and 1 additional hemolyzed lot. Each lots were spiked at low and high QC levels and analyzed to assess potential ion suppression and enhancement. The results summarized in Table 4 were highly consistent across all 7 lots, including the hemolyzed serum. This shows that the assay response for HTKIT was stable and not impacted by any endogenous component or sample conditions. Overall, there was no significant ion suppression or enhancements observed. As shown in Table 5, the stability of HTKIT was evaluated under various storage and handling conditions. The analyze remained stable for up to 28 days at both -20 degrees Celsius and -80 degrees Celsius for at least 23.8 hours at room temperature and after 4 freeze cycles. This result demonstrates that the assay is reliable and the sample integrity is well maintained throughout typical handling and storage conditions. In addition, several other evaluations were performed, as summarized in Table 6. The method showed excellent specificity with no significant interference in 6 screen blank matrices, including 1 chimerized lot. Dilution integrity was demonstrated up to 100-fold, maintaining accuracy within 60% and the precision below 8%. Recovery was consistent across QC levels, indicating reliable extraction efficiency throughout the assay. Reinjection reproducibility was also confirmed for up to 189.7 hours at 4 degrees, supporting reliable sample and reanalysis. In summary, we developed and fully validated hybridization LC-MS method to quantify HTKIT in mouse serum. The assay only needs 25 microliters of mouse serum and provide accurate measurement across the range between 1 to 250 nanogram per ml. By using a full-length complementary capsule probe, we achieve excellent selectivity, a very strong single-to-noise ratio, and a high sensitivity. Overall, the method met all validation criteria and is well suited for preclinical evaluation of vivo morpholinos. And thank you very much for your attention, and please do not hesitate to reach out if you have any questions.